Good morning, good afternoon, or good evening. Welcome to the International Society for Biopharmaceutical Statistics, ISBS Webinar Series. I'm Thall from St. Jude Children's Research Hospital, and it's my pleasure to welcome you all. Today, we are honored to have Professor Peter Thall with us for the presentation. Peter Thall currently is Annis G. Sauriel Professor in the Department of Biostatistics at MD Anderson Cancer Center, where he has served as a professor and member of the science faculty since 1991, totally 34 years. He is a fellow of both American Statistical Association and the Society for Clinical Trials, and received the Owen Award in 2014. Dr. Thall is a pioneer in application of the Bayesian methods in medical research. He has published over 300 journal articles and book chapters in the statistical and medical literature, co-authored the 2016 Chapman and Hall book Bayesian Designs for Phase 1-2 clinical trials, and authored the 2020 Springer book, Statistical Remedies for Medical Researchers, and the very new book, 2024, Chapman and Hall book, Bayesian Precision Medicine. Dr. Thall has designed hundreds of clinical trials, presented 245 invited talks and 35 short courses, and served as associate editor for Journal of the National Cancer Institute, Statistics in Medicine, Statistics in Biosciences, Clinical Trials, and Biometrics. I personally was fortunate to attend Dr. Thall's lectures on Bayesian clinical trial design during my PhD training at MD Anderson. His insights and perspectives have had a long-lasting impact on my thinking and continue to influence my work to this day. Thank you all for joining. And now, without further ado, let's welcome Dr. Thall to begin his presentation. Yeah, please.
Okay, let me see if I can do a share screen here.
Yes.
Share. Okay, can you see my screen?
Yes, perfect. Thank you.
View full screen. There we go. So thanks very much for that introduction. So this is kind of a topical talk that moves around. It's got a general title, Novel Bayesian Designs for Dose-Finding Trials, an overview, opinions, and examples. So let's jump right in. There we go. So I love this quote from John Kenneth Galbraith. The conventional view serves to protect us from the painful job of thinking. And I think everybody who does biostatistics should bear this in mind when they decide what to do or what not to do. So here's a brief history of dose-finding designs. And whoops. So the 3+3 design. For a new cytotoxic agent, you pick a dose using a very naive algorithm based on dose-limiting toxicity. Then the big advance, 1990, the continual reassessment method. What do? You specify a fixed target for the probability of dose-limiting toxicity, 0.25 or whatever you like. Then you identify a dose with estimated probability of DLT closest to that target. Then FTOX came along and we said, well, let's combine phases 1 and 2. So you drop a dose if its probability of toxicity is too high, or if its probability of response is too low compared to fixed limits that you specify or the doctor specifies. And you use a trade-off between response and toxicity to choose the dose. There are tons of elaborations of this. You can use times to toxicity and response, ordinal outcomes, optimize the dose pair, optimize dose and schedule, and on and on. And then the big step is to choose personalized doses. So now you've opened up the playing field to say, well, patients are not as alike as peas in a pod. If they've got different prognostic covariates, maybe they should get different doses. Another step is to use progression-free survival time explicitly. So response is not a surrogate for progression-free survival. And so the idea is to screen doses using the probabilities of something good or something bad early on, but then have longer follow-up and optimized dose using progression-free survival. Then there are all sorts of ways to combine the phases. So I've got a more illustrative way to show the history. So this is an illustrated history of dose finding designs. You see at the top, they began the 3+3, as far as I can tell, somebody cooked it up in 1950. Then the CRM came along 40 years later. You see the causal diagram is pretty simple. You give them a treatment or dose, the outcome's toxicity, that's it. And here's Dopey, he's a big proponent of that. And then, as we all know, dogs are far more intelligent than people. So a German Shepherd noticed that, well, you should also use efficacy as well as toxicity. And then Border Collies, which are smarter than German Shepherds, as we all know, said, well, let's also put in progression-free survival time. So these were the generalized phase 1-2 designs. And then Border Collies get together in different gangs, and they come up with all these different precision or hybrid designs, hybrid phase 1-2, phase 1-2-3 that use all sorts of different outcomes, toxicity, efficacy, biomarkers, time to event outcomes. So this is the way things have gone with dose finding. And here's some undesirable properties of conventional designs. I used to give a one-day short course in this. There are many 3+3 algorithms, and they all have terrible properties. So if you look at protocols and they say we're going to use a 3+3, you could collect, you might make a hobby out of collecting all the different 3+3 algorithms that they use, but most of them have many decisions unspecified. Unfortunately, they all stop with very small sample sizes, so regardless of what they did algorithmically, the final data are unreliable. And it turns out that any selected MTD has a very dispersed distribution from the small sample size. Therefore, it has high risks of being either too toxic or ineffective. The CRM, it turns out, if you think about it, is rather strange. Say you've got a CRM where the target probability is 0.25. So it's based on the idea is that the doses with toxicity rates closer to 25% are more desirable. One implication of that is, well, two doses with 45% and 5% toxicity rates are equally desirable, as insane as that sounds. I could talk about that for an hour. And then running a small, underline the word small, phase 1 trial is terrible statistical practice. Somehow people put their brains on hold and say, well, we can ignore the elementary principle that the reliability of any estimator or decision increases with sample size. We'll pretend that a sample of size 18 is just fine. And then the CRM, the 3+3 algorithms in the CRM all rely on an unstated assumption. And the assumption is that the probability of efficacy increases with dose, but they all ignore efficacy. And then there's the idea of expansion cohorts. They say, well, we've picked a dose, let's treat 30 more patients at that dose. So you've got an unreliable dose, and now you're going to treat 30 or 40 more patients. What if it's too toxic? Okay, so here's some, this is out of a review paper that we published a year or two ago. And so here's some actual instances where there was a trial that went wrong. And so there was a trial run in Philadelphia positive leukemia, otherwise known as CML, chronic myelogenous leukemia. They studied ponatinib and they used some 3+3 algorithm. It turned out after the trial, the dose they picked was unsafe. What a surprise. The solution is to include a rule to monitor the probability of toxicity. And there are lots of toxicity monitoring rules out there. The next disease was non-small cell lung cancer. They looked at something called arnatuzumab. Again, a 3+3 was used, and it picked an ineffective dose. What a surprise. The solution is include a rule to monitor the response rate to make sure it's not too low. And by the way, I should mention in passing, if you've got a toxicity-only phase 1 design and absolutely nobody responds, the algorithm doesn't look at response anyway, so it's going to ignore that fact. And at the end of the trial, it will pick a dose despite the fact that all the doses are ineffective. Going back to the examples, there was a trial in ovarian. They studied something called niraparib. They used some combination of accelerated titration and 3+3. It picked an unsafe dose. Well, here, you could use the utility of toxicity in response and also monitor the probability of toxicity to make sure it's not too high. Here's a trial that I actually designed. Poor little me. It's advanced leukemias. Barinostat was studied, and I used the tight CRM, thought I was terribly clever. It ended up at the end of the trial, and I'll show you how this happened. It picked a dose with poor survival, and the sample size was too small. I'll elaborate on that. The solution, which should have been done during the trial, was to count for survival time and increase the sample size at the lower doses. Then there was a trial of clear cell renal cancer using sitravatinib, and we used the late onset FTOX design. So now we're getting smarter, and all this says is we're going to look at time to toxicity and time to response and use a trade-off. But it was unable to pick a dose based on survival because survival is not in the mix. And so the solution is elaborate that design by following patients for long-term survival or progression-free survival. These examples all came from a colleague of mine, Pavlos Misau, who's he's got a limitless number of examples. So here's some general recommendations. When it is both feasible and ethical, account for long-term progression-free survival or overall survival or remission duration. Include Bayesian sequential monitoring rules that drop an unsafe dose if the estimated probability of toxicity is too high, drop an ineffective dose if the estimated probability of response is too low, and that enroll an adequate sample size to do safety and futility monitoring reliably and to do final dose selection and make final inferences reliably. If you can, randomize patients among the doses so that you can compare them fairly in terms of probability of toxicity, probability of response, progression-free survival, whatever you like, or to select a both fairly in terms of a utility or desirability of the combined outcome of toxicity and response. You see, if these are binary, there are 4 possibilities for progression-free survival. And then the holy grail is to account for patient heterogeneity so you can personalize the doses. So a patient with covariates, denoted by Z, gets a dose that's tailored to their covariates rather than using a one-size-fits-all rule. And this turns out to be pretty hard to do. So here's a first example. This is a trial that I designed that we actually ran. It's a randomized pilot study of cellular therapy for acute respiratory distress syndrome. So they were using T regulatory natural killer cells for treating patients with COVID-19 related ARDS. It means you can't breathe and you're in an ICU somewhere and you're dying. So toxicity is defined as any regimen-related severe, grade 3 or worse toxicity within 48 hours of the NK cell infusion. So at the time, giving somebody natural killer cells to treat ARDS was very, very innovative, very unusual. Response was a hard endpoint. It said that the patient was alive and extubated at day 28. Extubated meant they had a breathing tube in them, and the breathing tube had to have been taken out by day 28, meaning they didn't need it anymore. So they originally wanted to use a 3+3 or something like that to do sequential dose finding, and I talked them into randomizing because there was no reason to believe necessarily that the probability of toxicity or the probability of response would increase with cell dose. And so the 3 arms were 10 to the 8th natural killer cells, 3 times 10 to the 8th natural killer cells, or standard of care. And it was I constrained the randomization to have exactly 15 patients per arm, and it also was balanced to have 5 + 5 + 5, 10 + 10 + 10, and so on interimly to apply the safety monitoring rule. And here, let's denote the probability of toxicity by Q. Assume it has a beta 0.4, 0.6 prior, which means that the expected value is 40%, but the effective sample size is 1, so it's not very informative. And here was the stopping rule. This says to stop early if the likelihood that the probability of toxicity was above 40% was high. So if there was a bigger than 0.9 probability that the toxicity rate was bigger than 0.4, stop the arm. This was applied to each of the NK cell arms. And we applied it when they had 5 patients, which is very quick, and when they had 10 patients. And then the rule was if both arms are terminated, or both NK arms are terminated, stop the trial. So here's a schematic of the trial. You can see it's terribly complicated. You randomize, but it's kind of radical. People don't do this in dose finding. But here's what happened. Here are the final data in terms of just the counts. So the arms are standard of care, 10 to the 8th NK cells, and 3 times that. Nobody had any toxicity, which is good because they're dying of ARDS, but responses were pretty good. We had 9 out of 15 for standard of care, 9 out of 15 for 10 to the 8th cells, but strangely it went down excuse me, 9 out of 15. It went down to 6 out of 15 for the higher dose of the NK cells. The number of deaths, there were 4 out of 15 deaths with standard of care, 2 out of 15 with 10 to the 8 cells, and then it jumped up to 7 out of 15. And so one observation about the final data is that the high cell dose arm with 3 times 10 to the 8 cells had the lowest response rate and the highest death rate. Well, they're small samples, so you can't overinterpret. And going over these data with the investigators, they were constantly overinterpreting the results and acting like the 15 was 150. And I would argue with them on a daily basis not to do that. Anyway, there's a paper on this published. So here's the vorinostat trial. So this is a cautionary tale so that, we made the mistake. You try not to replicate it. At MD Anderson, there was a phase 1 trial of allogeneic stem cell transplantation for acute leukemia. And there were 6 doses of vorinostat studied. The acronym for vorinostat is SAHA. Let's denote the doses by 1 through 6. And it was added to the standard preparative regimen of busulfan. It's intravenous busulfan plus fludarabine plus clofarabine. And it was published, 3 years ago. So I designed it using the time to event CRM with a target 30% toxicity probability. Toxicity was defined as something very severe. DLT was grade 4, which means life-threatening, or grade 5, fatal, non-hematologic toxicity, or graft failure within 30 days. Very hard endpoint. But they didn't observe any. So the tight CRM did what it's supposed to do. It rapidly escalated to the highest dose level. And then this went on for several years. They just kept treating patients at the highest dose level behind my sleeping back. I didn't know they were doing this. And so here are the final per arm per dose sample sizes, 3, 3, 3, 4, 4. You can see it escalating. And then they camped out at dose 6 and just treated patients. And they didn't tell me they were doing it or show me the data. Okay, here's what happened.
Morning and good afternoon, depends on which time zone that you are. Welcome to Fuse Community Forum.
So there's somebody else is talking here. What's going on? Can you hear me?
I already mute, Doctor.
Okay. So what I did when they got done with the trial and said, okay, here are the data, I said, what happened? They said, well, we treated a lot more patients at the highest dose level. So I just drew some Kaplan-Meiers, and I reasoned that the sample sizes, the subsample sizes at the first lower dose levels were too small to draw Kaplan-Meiers, so I put them together. So the red curve is the Kaplan-Meier estimate of survival for the patients at the lowest 5 doses, and the blue curve is the Kaplan-Meier for the patients treated at the highest dose. You notice that it's lower. So the dose that got the most patients had the worst survival. And of course, these are not reliable curves. The implicit assumption here in plotting the 2 curves together is that somehow there was a randomization. That's not what was done. But the question is really now, what do you do? So one of the things that's often ignored in early phase trials is how do you make a decision based on the data from the completed trial? And the answer is, if you look at this, well, you really shouldn't use dose level 6. Probably you should use a lower dose level, but how do you choose among them, given that the sample sizes were so small? So you're sort of stuck, which is kind of a shame. So here is a phase 1/2, CAR and natural killer cell trial. So the goal of this trial, it's still ongoing, is to optimize the dose of CD70 chimeric antigen receptor natural killer in NK cells as targeted immunotherapy for relapsed or resistant B-cell hematological malignancies. And so you see the protocol was cooked up 4 years ago, and it's been amended so many times I've lost count. So the treatment is 3 days of chemo plus infusion of 1 of 4 doses of the NK cells. So it's 5 times 10 to the 6 or 5 times 10 to the 7 or so on. And just let's denote them by doses 1 through 4. So initially when they came to me, I just cooked up an FTOX design. So the idea was to treat up to 48 patients, have 16 cohorts of size 3 each. Toxicity is the indicator of grade 3 or 4 tox, means life-threatening, severe or life-threatening. Response was evaluated in 1 month. And a little notation, pi sub j of d theta. Theta is the model parameter, d is the dose, and j is either r or t. So pi sub j is the probability that outcome j, which is response or toxicity, occurred. So these are the response probabilities. Here's the standard safety monitoring rule. So the investigator said, look, the minimum response rate we'll accept is 50%. If it's likely to be below 0.5, then yes? I guess we have to mute something. If it's likely to be below 0.5, then the dose is not acceptable. It's inefficacious. And flipping it over to toxicity, if it's the investigator said, well, the maximum rate of toxicity is defined as 0.3. So if it's likely to be above 0.3, then that dose is too toxic. So these are rules for weeding out doses that are ineffective or unsafe. And then I had a conversation with a PI, a fellow named David Marin. And I looked at the protocol and I said, look, it only takes a month to evaluate response and toxicity. So why do you have follow up to 6 months? And he said, we want to evaluate response durability because many patients who initially respond to the NK cells relapse fairly soon. So I thought about it and it was like, oh, this is the old problem that you get a good or desirable early outcome, and you get the patient into remission, but it doesn't last. So that by month 6, either they've relapsed or they've died. So I thought about it. I said, David, okay, I want to redesign the trial. Here's what we came up with. So the motivation for this Gen 1-2, it's a generalized phase 1-2. Short-term toxicity, denoted by a red Y sub T, and response, red, blue, Y sub R, are never perfect surrogates for the long-term outcome of progression-free survival or remission duration, evaluated over, say, 3 or 6 or 9 months. So the idea is the evaluation window for the toxicity in response is short, and the evaluation window for PFS is longer. So a dose that maximizes a phase 1-2 objective function based on the early outcomes does not necessarily maximize any objective function in terms of the long-term outcome. Let's just call this for PFS. And so the observation is that, however, the early outcomes still provide information about the longer-term outcome. And this is just the law of total probability. So the distribution of the time to event outcome as a function of dose is just the average of the conditional distributions where you're conditioning on the early outcomes and we're multiplying by the early outcome probability. Just the law of total probability, nothing deeper than that. So the idea here is that, look, the early outcomes are not perfect surrogates, but there is a conditional distribution of the long-term outcome that does depend on the early outcomes. Okay, so how do we exploit this? So here's a toy example. The Y sub R, early response, and PFS time may disagree. So let's assume for simplicity that the agent's safe so we can ignore toxicity. So this is just for illustration. And say we've got doses D1 and D2. And we'll denote non-response by 0 and response by 1. And I'll say P12 of D and Y is shorthand for the probability that the PFS time is at least 12 months, given dose and the early response outcome, 1 for yes, 0 for no. So here's a table that I cooked up to show you that probability can be counterintuitive. So in this toy example, for dose 1, the response rate is 0.2. For dose 2, the response rate is 0.4. But when we look at these conditional probabilities of surviving 12 months, or PFS longer than 12 months, of 0.95 for responders, dose 1 has 95%, dose 2 has 75%, and we have different numbers, 85 and 65. When you just crank out the solution to the probability computation, you get that the 12-month, the unconditional 12-month PFS probability for dose 1 is 0.87, but for dose 2 it's 0.69. And so the point here is simply that if you look at early response, dose 2 is much better. It's got twice the response rate. But if you look at the 12-month success rate in terms of PFS, dose 1 is better than dose 2. And so this is a very simple example, and all it says is that what happens with response in terms of picking a best dose what happens with long-term PFS don't have to agree. And very often they don't. We actually had a trial where this happened, and that's a whole story unto itself. So here's the Gen 1-2 design. It's a new paradigm for early phase designs. And so in stage 1, you pick your favorite phase 1-2 design, whatever you like, but it's got to have some sort of objective function based on the early outcomes. And then you use toxicity in response to screen the doses. And here's the line of demarcation. We choose a candidate dose set. So rather than picking one dose, we choose a set of candidate doses, which if you start out with 4 doses, the candidate dose set may be 2 or 3 or something like that, or 6 doses. You don't want a candidate set to have more than 2 or 3 doses. And then here's the big step. You randomize more patients among the candidates, follow them a bit longer for as long as it takes to evaluate PFS, and select the best dose based on the PFS time, the Y sub S. Now, Gen 1-2 is actually quite general. It can be used to choose an optimal dose, an optimal schedule, an optimal dose schedule combination, and whatever rings your bell. It's up to you. It's just a paradigm. So here are the elements of the design. I'll denote toxicity in red by YT, response in blue by YR, YS is remission duration, or PFS time, or survival time. Now here are the 2 follow-up times. T1 is the short time, which usually is like 1 to 2 months to evaluate response and toxicity. T sub 2 is the longer follow-up, which is 3, 6, or 12 months to evaluate the long-term outcome, the time to event outcome. And model parameters are denoted by theta. So here are the two decision criteria functions. And so the phi of dose D in parameters theta is some sort of short-term phase 1-2 objective function. It can be a mean utility, an efficacy toxicity trade-off, an odds ratio, whatever you like. The psi of d theta is a long-term success criterion. And here it can be the expected progression-free survival time at that dose, or the probability that the PFS is bigger than T2. So as in our example, maybe the probability that Y sub S is at least 12 months. So here are the 3 stages of the design. Let's let lowercase n 1, 2, and 3 be the per stage sample sizes. So when you add them up, you get the total sample size. And then let's break it down further. N sub S of D is the number of patients treated at D in stage S, 1, 2, 3. So for stage 1, you just conduct a phase 1-2 trial with N1 patients, choose a dose for each cohort to maximize phi as you do, choose drop doses that are too toxic or inefficacious. So everything so far is just a phase 1-2 trial. But at the end, instead of picking one dose, you pick a set. I'm going to write it as calligraphic C of candidates. So now that you've got a candidate dose set, now we've got our let's treat additional patients rule, adaptively randomized N2 patients among the doses in the candidate set. And there you can do a little mini simulation to say, well, how big should N2 be? And so you try different values of N2, should N2 be 5 or 10 or 15? The bigger it is, the bigger your trial gets. So it's got to be, something the investigator can afford. And you see how reliably you can make decisions. We found that values of N2 of 10 tend to work well. And then you follow each patient for T2 months, maybe 6 months, and then use your long-term criterion to select a dose. So select a dose to maximize the estimated probability of long-term success. So here's how we've kind of snuck long-term progression-free survival or remission duration or whatever you want into the dose-finding paradigm by saying, let's extend phase 1-2 a little bit. Now, to identify a set of candidate doses, let's denote the data after N patients by calligraphic D sub N. Calligraphic A is the set of acceptable doses. And then phi hat sub JN is the expected value of your short-term criterion for dose J. And this is what you would use to pick doses if you're just doing phase 1-2. And then the AR probabilities, you can do it any way you like. You can make them proportional to this estimated early success criterion raised to some parameter. The tuning parameter can be 1 or 0.5 or something like that. And so the candidate dose set, if you look at this expression, in words, all this says is to be in the candidate set, the estimated optimality criterion for that dose DJ has got to be at least rho times the maximum value. So the maximum value is what you would ordinarily use to say, here's the best dose. But rho is a value less than 1. So if you say rho is 0.7, what you're saying is, well, if you're at least 0.7 or 70% of that maximum value, you get into the candidate set. And we played around with this and tweaked it. And you can look at 0.5, 0.6, 0.7, and so on and see what works. And this is because, obviously, when you're picking a candidate dose set, you've got rather variable data. This says that, well, you don't really know that the dose that maximizes the short-term criterion is best. So let's open things up a little bit so some candidate doses can sneak in there. Now, for the long-term outcome futility rule, Yang Zhang put this into the design, and it really is important. A candidate dose has to satisfy a long-term futility criterion. So for the long-term success criterion, it's got to have at least a 10% chance of being at least some fixed lower limit psi. And for example, if psi is 0.4 and the long-term outcome, the long-term criterion is the probability that PFS is at least 6 months, what this is saying is, look, the probability of a 6-month success rate being at least 40% cannot be vanishingly small. It's got to be 0.1. And what this does is keep you from including candidate doses that just don't have much of a chance of long-term success. So if, based on the long-term follow-up data, no dose has at least a 10% chance of achieving a 40% 6-month PFS probability, then no dose will be selected, regardless of how promising the phase 1/2 results were. This has to do with the disconnect between the early outcomes and the long-term outcome. However, if one or more doses achieve high early response rate, but the responses are not durable, then no dose will be selected. So a negative phase 3 trial is less likely to be conducted. So this rule is a gatekeeper. It says don't waste time and resources on a dose that's got early outcomes that look promising, but has, not so good outcomes in terms of long-term success. So if you're in the setting where a lot of patients get responses, but almost nobody is alive with disease in remission at month 6, don't proceed. Don't go to phase 3. So this is a gatekeeper rule. So planning stage 3 adaptively, there's this whole business that we go through where we say, well, n sub 3 of D is the stage 3 per dose sample size, and it's usually 10 or 15 or 20 or something like that. And the way we pick it is we do a little mini simulation. We simulate stage 3 for assumed values of the true objective function. So now you're in the middle of the trial and you're saying, well, I've got my data from stages 1 and 2. What is stage 3 likely to look like? Well, you take the data that you've got so far and you say, well, what are my criterion functions, the psi values at each dose of the long-term criteria, and the phis at each dose or the short-term criteria. Let's just simulate the trial for different values of those and see how it behaves. Use that to pick our per dose sample size in stage 3. Here's a little mini example. You've got 4 doses. Your candidate set turns out to be doses 3 and 4. You've already treated 12 patients at dose 3, and you've already treated 6 patients at dose 4. And so if you say, well, I can afford 20 patients per dose, then through the magic of subtraction, the stage 3 sample size at dose 3 is 20 minus 12 or 8. And the stage 3 sample size at dose 4 is 20 minus 6 or 14. And then you say, well, I can have 20 more patients. So what is 6 and 14? So I made a mistake here. One of these is, this should be a 6. So this should be a 20. I made an arithmetic error. This should be 6 plus 14 is 20. You randomize these additional patients between dose 3 and dose 4, restricted to have the values that you, excuse me, 20 patients per dose. Yeah, I made an arithmetic mistake here that I can't recover from. So I think what I did my example is if n of D is 22. So I apologize for that. I didn't pick it. So if n of D is chosen to be 22, then you could have 8 at dose 3 and 14 at dose 4. And so you'd randomize them restricted so that you had 8 and 4 at dose 3 and 14 at dose 4. So as they're arriving in the clinic, you're randomizing them to get these sample sizes. I'm sorry, the 20 should have been a 22. And then when you're done, so this is this whole elaborate thing for, well, how do you pick the subsample sizes at stage 3? When you get done, you've got estimates of the long-term success criterion at each of the doses, and the larger one wins. So that gets you to the long-term outcome. So now here's a utility-based Gen 1/2 design for the natural killer cell trial. And so here, toxicity it occurs or it doesn't, but response is scored as a 3-level ordinal variable: progressive disease, stable disease, response. So RES is really CR or PR. So there, 2 times 3 is 6. You have 6 elementary outcomes. So here's the assumed utility. The utility of progressive disease with toxicity is 0, because that's the worst outcome. The utility of response with no toxicity is 100. That's the best outcome. And then the utilities in between for the intermediate outcomes, you get those by talking to your clinician and say, how desirable is this combination as a value between 0 and 100? So it's completely subjective, but it tells you what the trade-offs are. So let's denote the joint probability for outcome A, which is 0 or 1 or 0, 1 or 2. It's 0, 1 or 2 for progressive disease, stable disease response for Y sub R, and likewise B has 2 values. Now let's take this vector of joint probabilities and just string it out as a vector of P0, P0, all the way to P1, There's 6 probabilities in there. And make a very, very, very simple assumption that if you record the 6 vector of counts for this, that it's multinomial, and the parameters are n and that probability vector, and give it an equally likely Dirichlet prior. This is a very simple-minded model that doesn't assume much of anything. It's like, well, if you have 2 outcomes, it's a beta. If you have 6 outcomes, it's Dirichlet. And then let's the long-term outcome be remission duration, and assume that it's Weibull. I should mention there's a very nice paper that was published recently where these people looked at a ton of medical papers with different data sets to see how robust are the Weibull and log normal. They found out that the Weibull or log normal fit almost anything. It was very amusing. After all the years of goodness of fit, it turns out if you're willing to assume a Weibull distribution, you can fit all sorts of data sets. Anyway, let's assume that remission duration follows a Weibull, and the rate parameter is a function of toxicity. So you've got a bump beta sub t for toxicity, and then the gamma j is the bump for them getting dose j. So beta t, toxicity effect on remission duration, gamma j, dose dj effect on remission duration. So then here are a bunch of parameters. And so we noodled around with these. And so if you have cohort of size 3, N1 is 15, N2 is 33. It's just a particular case. And that adaptive randomization parameter is 0.5. Now, our row of 0.7 says that to be in the candidate dose set, your estimated criterion, short-term criterion, has got to be at least 0.7 times the maximum value. That gets you your candidate set. The short-term follow-up is a month. The long-term follow-up is 6 months, which is very, very common. The long-term success is it's the event that the long-term remission duration is at least 5. This says, well, you scored their response at month 1, and so do you go to month 6 and still be is the patient still alive and in remission? So 6 minus 1 says their time to loss of response, to fall out of response or to die is at least 5 months. And this is given that they're a 1-month responder. So it's a conditional probability we're going to compute. So stage 1 is just a kind of vanilla utility-based phase 1/2 design. So the criterion is the mean utility. You just take the utility function and average it using the probabilities. Remember, these were Dirichlet. And so the theta vector is that Dirichlet probability vector. And so if you were just doing a phase 1/2 trial, stage 1 would be everything. You'd optimize that. Stage 2 says, no, let's continue. We'll adaptively randomize up to N2 over C, or N2 over 3 more cohorts of size 10 each among the acceptable doses, and then identify our candidate set. as I described earlier. In stage 3, you randomize N3 patients fairly among the candidates. So now once you've done that, you just evaluate. Your optimal dose using the long-term criterion is based on your final data. And so here, in this particular configuration, the expected value of the long-term criterion given the final data at the end of stage 3 is what? This is the probability that your remission duration was at least 5 months. And of course, the times can be adjusted to whatever the investigator thinks is reasonable. So they were evaluating response at month 5, and then remission duration at month 6. So you get the survival probability at month 5 as a function of dose and parameters. And so since we're Bayesians, we take the expected value of that given the data. So that's the trial. Okay, so here's a schematic of the design. So this first thing with all the curly Q arrows is just phase 1-2 dose finding, and you eliminate unacceptable doses. Then the big arrow going down says, then you randomize and select. So you randomize and select a candidate dose set. Here in this schematic, the candidate doses are doses 2 and 3, and you continue to randomize. You eliminate unacceptable doses. You continue to do that. And the follow for progression-free survival or remission duration, whatever it is you want. And in the end, it turns out dose 3 was selected as optimal based on the long-term outcome. So that's that. We simulated this a lot. So here are 2 conventional comparators. The first conventional design says, look, let's just do a phase 1-2 trial with 2 stages. I mean, that should be good. So it has the same stage 1 and stage 2 of the Gen 1-2 design, but then it's done. It says, here's an optimal dose, and it goes home. There's no stage 3. So that's the kind of vanilla phase 1-2 design. The conventional design 2 is nearly identical to the first one, but it randomizes more patients in stage 2 so that the sample sizes match Gen 1-2. So the first conventional design on average would have fewer patients because there's no stage 3. We say, well, to make it comparable, let's let this conventional design have more patients so we get a more fair comparison. And here's what we got. So the first simulation scenario, it's got no acceptable probabilities of toxicity or response. And so you don't want to pick anything. And so what I've given here in boldface are the optimal decisions. And the optimal decision is to pick no dose. And so you see that the Gen 1-2 design does that about 94% of the time, but the conventional designs do it far less frequently. So the conventional designs are not very good at picking, saying no dose is optimal, because the conventional designs aren't smart enough. That is, they don't have this additional information on the long-term outcomes. So if we didn't get this in the simulations, we'd know something was wrong. So this is sort of the obvious case. If you look at long-term outcomes, you're gonna have an advantage. But remember, when most people do a phase 1-2 trial, they say, oh, isn't that wonderful? And they pat themselves on the back. And then they sit around waiting for the data for longer follow-up and see what happens by month 6 follow-up. And you can get the embarrassing result that, well, your best dose didn't have very good long-term success. So this avoids that. In simulation scenario 2, no dose has acceptable probability of toxicity and long-term criterion. So here, the Gen 1-2 design picks it stops and says nothing is acceptable 84% of the time. The conventional designs almost never do that. They just don't. And so again, the unacceptability, the doses being unacceptable is because their toxicities are too high as you go through the different scenarios, or their long-term success rates are too low. So you see the psi true of DJ. There, the limit on that was 40%, and these are at 0.1, 0.15, and so on. And so if you're saying, I want at least a 40% long-term success rate, but they've actually got rates of 10 or 15%, you don't want to pick any of those doses. So this is a different criterion that doesn't exist in a phase 1-2 design, because the phase 1-2 design does not have a long-term outcome. So again, if this hadn't been, we hadn't gotten results like this, we'd say something was wrong. But here, I was really surprised at the immense difference So the conventional designs just crash and burn. So here's a scenario where dose 4 is best. You've got a good dose. Okay, so if you look at the mean utilities, you say that, well, the mean utilities are 61, 67, 74, and 75. And the true long-term success rates are 0.2, 0.4, 0.5, and 0.7. Remember, our lower limit on that is 0.4. So clearly the dose 4 has got the highest utility by a smidge over dose 3, but a far higher long-term success rate. What are the selection rates for dose 4? Well, Gen 1-2 selects it 70% of the time, but the two conventional designs selected 30% of the time fewer. So again, it's because the conventional designs don't have a psi true. They don't see the long-term outcome. So they don't have this criterion in their structure. And then in simulation scenario 4, doses 2, 3, and 4 have got ideally identical mean utilities, but dose 2 has a much higher long-term success rate. So this says, look, if the mean utilities jump from 54 up to like 72, or was it 73 to 78, so doses 2, 3, and 4 are a lot better, and dose 4 is the best. But your long-term success rate is 70% for dose 2, which is much bigger than the other ones. Then the problem of picking an intermediate dose is always hard. And here it should be done in terms of long-term success. The Gen 1-2 design does it 68% of the time. And the conventional designs, they fall all the way down to 24, 25. So again, the long-term success criterion makes a huge difference. So the advantages of a Gen 1-2 design are you get better dose optimality criteria because you're looking at a long-term outcome. It's very practical, and the reason is it's kind of trivial. It's from my conversation with David Marin. If they're going to follow the patients for 6 or 12 months, take advantage of the data. That's all that I'm saying. You've got the data there, use it. It's very general and flexible. You can look at dose and schedule. You can do whatever you like. You've got less anxiety. There's no need to worry about having to admit, oh no, we chose the wrong dose. So I think this is an advance over what we typically do if we do phase 1-2. And if you go all the way back to 3+3, those people are basically in the Stone Age. So here's a new design I thought I'd toss in at the end. It's ROMI. Everybody likes acronyms, so this is for randomized, optimal, two-stage basket trial design for multiple indications. And the outcomes are response and toxicity. Here, the setup is very simple. You want to identify at the start 2 doses, a low dose decibel and a high dose decibel. And these could be, for example, an MTD and a recommended phase 2 dose. And so you've got those 2 doses. Can you choose between them? But what we're doing here is looking at 2, 3, or 4 indications. You've got several different diseases. You may have a couple of them. You may have 4 of them. And the motivating trial is an anti-CD137 agonist, plus something called pembrolizumab, plus nab-paclitaxel for metastatic solid tumors. And the doses of pembrolizumab plus nab-paclitaxel are fixed. So we're varying the doses of the agonist. But the doses of the agonist were either a high dose, which is the MTD, or a low dose, which is the MTD -1 level. And Yin Yuan is the one that formulated this because his reasoning was, look, this situation comes up a lot. You come out of a trial and they give you these 2 doses, and how do you choose between them? And very often it's an MTD or an MTD 1 step down. So here are the indications. There was esophageal gastric cancer, head and neck cancer, HER2 negative breast or ovarian. So you see you have 4 very different indications where we're trying this out. And you want to allow yourself to make a different decision within each indication. So this is an example of precision medicine. So in stage 1, you want to screen for the higher dose. So you run a small single-arm trial with D-sub-H in each indication, and then you drop it if it's too toxic or inefficacious. And so the specifics for this is sort of obvious. And if you drop D-sub-H in an indication, then stage 2 is not conducted, and that's that. Otherwise, you go to stage 2. So in each of the indications, you could either just stop early and say, that's it, we're done, or say, well, no, this higher dose gets moved to the indication so we can have stage 2. So in the stage 2 dose optimization, you only get there if your high dose was found in indication K, if your high dose was found to be acceptable. And the goal then is to optimize between the low and high dose. So it's once you're within an indication and you're in stage 2, there are two decisions you can make. Which one is better? Or you could say, well, neither of them is any good. We found they're too toxic. So for each indication, now you randomize some more patients between the low and high dose. And now we're going to do a utility-based decision-making. So what we define is, this is kind of cute, I think. You get the counts of each response and toxicity pair. So there are 4 combinations here. So you've got 4 of these different X values. And then you use those to get a utility-weighted count. It's a utility-weighted average count. You multiply the utility by the count, add them up, divide by 100 to get something between 0 and 1. Now you can use this cute quasi-binomial formulation on this X bar that you've concocted in indication K. Okay. And so the L is low dose, high dose. And then one thing that we do to reduce dimension is cluster indications that have got similar distributions using latent clustering variables. So that's a cute thing. You don't have to do it, but it saves some makes things a bit more reliable if you have 2 diseases where the dose outcome models are similar. And then with each cluster, you cluster meaning indication or combined indications, you just pick the optimal dose between the low and high dose to maximize posterior mean utility. So with all the structure, when you get done at the end, you can pick which of the 2 doses you like better. So here's a schematic. The Shuki Wang cooked this up, and I'm not going to go through the whole thing. It shows you got 4 indications, and you do screening, and in this you've got, well, indication 1 there wasn't anything any good, but indications 2, 3, and 4 have at least got something. Now you go to stage 2 where you randomize, and so I won't go through the rest of all this. Anyhow, that's my talk for the day. Oh, excuse me. We've got this is my talk. All I did was write down the utility functions for the indications. The point here is the utility functions for the different diseases don't necessarily have to be the same. So if you had, a homogeneous sample with one disease, there'd be 1 utility function. Here, there could be 4 utilities. Okay, so thanks for your attention, and here's some references. So if you're into reading papers, you can, if you don't have the slides, I can send them to you, and you can read all the stuff that we've published lately.
Thank you very much, Dr. Thall. And any questions from audience? Okay, maybe I have a very quick question in the first place. So thank you very much for this gene 1-2 design. And I think people may be wondering if there is any user-friendly software available for others to want to apply your method?
Yeah, well, for the Gen 1-2 design, the once it did the coding were Yang Zhang and one of his younger colleagues. So there's code sitting out on a website. So if you go to the paper, where is it here? Yeah, this paper in Pharmaceutical Statistics.
Okay.
There's a link there where you can get a computer program. And if you try it out and you have some problems, then please give us some feedback so that we can improve the code. Because as people that have been down the road with this sort of thing know, there's a lot of parameters and there's a lot of stuff calibrating the prior. So there's an awful lot of stuff that you've got to do specifying design parameters and hyperparameters. You've got to do a lot of simulations. So if you have trouble with that, please communicate with us.
Great. And I think another question is that in current practice, a common and sometimes ad hoc approach is to complete a phase 1 or phase 1-2 trial based on the short-term efficacy endpoint, and then usually in the pharma or the cancer center, and we decided on the next steps. Which kind of the dose or doses should be explored based on the totality of evidence and a lot of discussions after this kind of stages.
Yeah.
Yeah.
Go ahead. I'm sorry.
Yeah, sorry. And okay, and based on and after this kind of the short-term efficacy endpoint, this trials is done, and we will see which dose or doses should be go to next stage, right, to assess longer-term endpoint. And currently, your proposed GN12 design formalize this process into the single integrated framework.
Yeah,
However,
that's
yeah,
exactly
yes.
exactly what it does.
However, yeah, because I just want to see that this also introduce a little bit operational challenges, and because there is a lot of, we will make a decision, and many times we should lock the database, etc., this kind of things. I just want to, can you comment on that kind of, I don't know, it's can called trade-off between statistical rigor and real-world feasibility?
Oh, this is a great question. So yeah, I think that whoever wrote that down noticed that what we've done is attempt to formalize what people do informally when they're going through the dose funding process, and they say, well, we did phase 1, 2, we've got longer follow-up data, here's what we saw, and then they decide what to do next. And so all we've done is say, well, let's formalize it by saying the longer follow-up is for a 6-month outcome, say. And so if you incorporating that in the design ahead of time, that is, if you say, as David Marin did it, we're going to follow all of the patients for 6 months. I said, well, if you're going to do that, I'm going to embed that in the design. So logistically, my notion of things was, if they're going to follow them for 6 months, I want to take advantage of the information that's there. Now, if somebody says we're going to follow them for 9 months or 12 months, and you can still do it, but you're going to have to wait longer before you pick your dose. So I think that in applying I've applied the Gen 1-2 paradigm, and it's got several different versions depending on the constraints imposed by the clinicians I work with. And the trial, and the CD70 trial now it's really got multiple indications. They're trying out these NK cells for solid tumors. And so it's a really high bar using cellular therapy to get responses in solid tumors and make them durable. So it's a very hard trial. And they keep changing their mind. So I've gone through 3 or 4 iterations of the design with them. So I think as people try out the Gen 1-2 that puts it all together, they may discover, well, this is feasible, this is not feasible, and so on. And so you only know that once people have tried it out. But I think that one of the truisms about early phase trials is that we've got a culture that's deeply embedded that says we don't want our early phase trials to be very big, because they're just to pick a dose. And once we've got a dose, now we can decide, well, is the response rate high enough, et cetera. And then we can decide whether to go to phase 3. And phase 3 will be a great, big, huge trial. So I've got 2 contentions about this. One of them is our early phase trials are not big enough. We should have a cultural shift so that more patients are being invested in early phase optimization of dose or dose and schedule or what have you, so that we've got more, and they should be followed longer, so that, and I didn't include this in the talk, we can compute predictive probabilities of phase 3 success. And the reason I think this is important is that many phase 3 trials are begun that end up giving negative results. But my contention is that could have been predicted with reasonable reliability before they started the phase 3 trial. So the thing I didn't talk about at all today that's appended to this is the use of predictive probabilities for finding out whether phase 3 is feasible. And to make things even worse, we've come up with a all-in-one phase 1, 2, 3 design where the prediction and the phase 3 trial are included in the whole regime. So if you take this Gen 1-2 design and you say we may or may not go to phase 3, depending on how our predictive probabilities come out, now you've got the whole thing under one umbrella. And somebody could look at that and say, wow, it's even worse. It's just too much to commit to at the beginning. So the difference is, do you do things a step at a time, doing one, maybe just phase 1 or phase 1, 2, look at your data and then decide what to do next? Or do you put it all together and say, well, here are the decisions we might make. Let's put it all in one design. And that's harder to do. But I think that once you're simulating the whole thing and you see what happens, one of the things to notice, if you go back, is this. So this is a complicated scenario where you've got 3 doses that are okay, but dose 2 is the best. And you see the Gen 1-2 design does a far better job of picking that best dose than the conventional designs. So conventional means some sort of phase 1-2 design. You see that was true in the other simulation cases where the Gen 1-2 design just does far better. So the kind of meta fact about this is if you're able to incorporate this longer term outcome in the design at the start, you're going to get far more reliable results. And that's all I know about it at this point. And so to use this, you're going to have a cultural shift, certainly away from 3+3, but I think beyond on phase 1-2.
Yes, great. Thank you very much. I think currently already 11, we have 3 questions on the chat. And the first 2 questions is about just ask, could you give some intuition about how the utility functions are defined in practice? And another question is that what are your thoughts or suggestions if any, on incorporating PK information during those finding studies? And yes, how this can be done on the design stage.
Everybody says, well, you should ask the patient. So the patient has not gone through this treatment before, so they don't know. And if you say, here are the 6 possibilities, what are your utilities? And they'll say, well, I'll give 100 to no toxicity with response and 0 to everything else. Because the patients haven't gone through the process and they don't know how desirable or undesirable these things are. However, the physicians have treated lots and lots of patients. So what I do, just pro forma, is I nail down the 0 for the worst outcome. I nail down the 100 for the next outcome. And then I show it to the clinician or a group of them and say, what numbers do you assign that quantify how desirable the interim outcomes are? And they'll just give you numbers. So what I've found in practice is that eliciting utilities from physicians is very, very, very easy. They love to give you their utilities. They love to do it. But that's not a problem at all.
I think the last question from Demetrius is, any thoughts on suggestion, if any, incorporating PK information during those finding studies and the design stages, and it is worth effort in practice?
Yeah, because PK data takes a while to generate. That is, you've treated them, and then you've got to do these PK studies to find out what the variables are for each patient. So now you've got an expanded data set, and the question is, well, can you personalize things based on the PK outcomes, et cetera? And I think it's a great idea since you're going to follow them longer. You've got long and make adaptive decisions later on. It makes sense to me to extend this to use PK data from the previous patients. But of course, you're going to have to elaborate the model. You're going to have to say, well, response and toxicity, the probabilities depend not just on dose, but on the PK data. So yeah, it's a terrific idea, but it's going to require some very delicate modeling, I think.
Hey, yes. Thank you very much. Thank you very much, Dr. Thall, and very many insightful, thought-provoking presentation and discussion. We truly appreciate your time and expertise. And thanks also to everyone joined today. And yes, I think we can conclude today's ISBS webinar and looking forward to seeing your future events. Have a great day or evening. Thank you, Professor Thall. Hope to see you soon. Yeah, thank you very much. Bye.